On behalf of Cambridge Health Tech Institute's Breakout COVID-19 Global Web Symposia Series and our sponsor, LARVOL, I'd like to welcome you to Translating Biomarker Literature into Novel Insights. My name is Elizabeth Lamb, and I'm the host and technical director for today's event. Now I'd like to introduce our moderator for today. He is Mark Gramling, PhD, Director, Oncology at LARVOL. Welcome, Dr. Gramling. The presenter ball is yours.
Excellent. Thank you, Elizabeth. All right. So we'll get going here. Thanks all for joining and those who may still be filing in the proverbial virtual room. So thank you for that introduction, Elizabeth. So my name is Mark Gramling, and what I'd like to do today with my co-panelists is give you a 10,000-foot view and sort of open a forum for discussion about what my team is building at LARVOL, focused specifically on the biomarker literature and how we can utilize both technology tools as well as curation to bring all of that literature and parse it and index it and bring it into a platform that enables users for both research and drug development purposes to stay abreast of the biomarker literature. With that very broad self-introduction, I'd like to take the opportunity here to introduce my co-panelists. So we have Karna and Vincent, and I'd actually like for them to introduce themselves now. Whomever would like to go first, gentlemen, please go ahead.
Okay, I'm Vincent. I'm very happy to participate to this webinar with you, Mark, and Karna. I enjoy using all the LARVOL applications. So I'm a drug discoverer. I have over 20 years of experience in drug discovery, working with all kinds of modalities. And I'm right now a senior director of medicinal chemistry at a company named Protagonist Therapeutics in California.
Hello, this is Karna Sheth. Welcome, everyone. Thanks for being here. I'm the principal consultant of my own firm, Integra Connect, LLC, based in Chicago. And I've been working with a lot of real-world oncology data, helping both practices and payers find opportunities to improve outcomes and reduce cost burden. So it's great to be here, and thank you, Mark, for having me.
Thank you, Vincent. Thank you, Karna. Yeah, it's a pleasure to have both of you here with me. Vincent, from maybe that drug development perspective especially, and being a power user of some of the other LARVOL products as well, and invited Karna to be with us today, bringing a different perspective on, there's a lot of parallels with what we're honed in on public data versus, what are the similarities and differences with, patient data and real world data. So that's oft the first topic of conversation when we focus in on what we're doing with Very, which is a public data sort of domain platform. So we're going to get going. I'd like to give first a brief introduction of LARVOL, very brief, and then get into just some basic slides about the different components of what we're doing in biomarkers, and take the discussion from there. So just as a broad setup to familiarize with LARVOL Group and what we do. So for the past 17 years, we've been building customized intelligence reports and software as a solutions primarily focused in the pharma industry, but also in biotech and some healthcare as well. So these SaaS solutions have been inclusive of alerts about drug pipelines and mechanisms of action, conference planners, tracking of key opinion leaders, and a variety of platforms in that space. But what we wanted to hone in on in the past year, and what I was tasked with building a team to do, is start to focus in on oncology and how we can expand those tools and expand those approaches to hope to bring some insights in both oncology predictive biomarkers, but also in other spaces. But I focus primarily on the biomarker element, and it's been a fun ride, leaning back to my original days studying cancer pathology in breast and lymphoma, and now being able to explore a lot of different tumor types. So that's the broad strokes of what we're doing at Very. So with that said, now let's maybe start digging into what we do, what our approaches are, what our data sources are, and see where questions might come from that. And I'll level set with everyone. I'll do my best to keep track of the comment section for everyone here. So this first question is fantastic, I think, coming in about validation. So let me set up just broadly the framework, and it's a fantastic question. So in broad strokes, what initially wanted to set up was, being able to take all quote-unquote different levels of evidence, from the highest regulatory approvals down to even the earliest preclinical studies, and be able to apply text mining as well as curation to bring them into whether it's an email-based alert package or the platform, as we'll see later. But to speak to the question just submitted is, what about the level of validation? And perhaps it's a unsatisfying answer to the question, but we code to the level of the publication. So we have erred on the side of being all-inclusive to have the data indexed in the database and thus linked to the primary source, because our job is to triangulate the user to the what we call the association, the predictive relationship, which is easier to see on the platform, and direct them to make their own conclusions from the primary data. We've decided not to jump in to be arbiters of that validation, and it especially comes up when it comes to conference data. We've discovered that for our pharma customers, we've erred on the side of bringing them value and the latest data from conferences, even though it's self-evident that that data can change. And so perhaps to a pause, I certainly don't want to turn into a speech. Is there any questions or comments from Karna or Vincent? Perhaps, the types of data frameworks they're used to working with, the types of data sources that they find most interesting. Obviously, Vincent is a user of the platform, so just wanted to pause there if there are other questions.
Yeah, I mean, definitely, I'm a drug discoverer. In general, when I use I'm looking, for instance, for a specific drug. So I have a name and I want to see exactly how it's progressing in development. If not a specific drug, I would look for a class of agent, like, I don't know, for instance, EGFR inhibitors or KRAS inhibitors, and we'll see which one are most advanced. I will definitely look for recent development. So obviously, tracking information coming from a scientific conference like ESMO, ASCO, AACR. That's definitely something I like to grab information from VI.
Yeah, I'll echo a little bit what Vincent said in terms of the governing bodies, ASCO, ESMO, NCCN. I'm always comparing what's happening in the community oncology setting to those guidelines as kind of the first pass standard. Also interested in the journals and the publications and updates on data. A little less concerned with the trials, since at least obviously in the real world, these are post-approved agents for at least something. So off-label use is somewhat interesting, but the trials are too small compared to some of the community oncology data trying to gather and review.
What I would add is, I know Mark will discuss that in the coming slide in this using the heat map. I'm definitely a big fan of the heat map because in general, when I want to see, obviously, for instance, in cancer, people will advance drug for different indication. In general, we look for combination, and I think the heat map is very useful for that, for looking how the people are trying to pushing forward in the agent, what combination they use for what cancer indication, etc. I think the heat map summarizes very quickly. I think it's very easy to use, and that's definitely a great tool for me personally.
Excellent, and appreciate those perspectives, gentlemen. I think we'll see once we go to the heat map. We'll see that the directive there is that we really index fairly flat data. What we index is, the tumor type, the biomarker, which we cover a variety of biomarkers, and the responsiveness to the therapy. So our job is to sort of create that. It's like a library, which we've organized into a heat map, which we'll go toward, and then direct the user. So there's noise and there's a lot of assessment to be had there. but that's what Vincent's speaking to, is a way to jump and move is what we're aiming for. But we'll definitely get maybe more of a sense of that, and I'll jump to a live demo when we go to the platform. But for sake of completeness, and because this is the order of my slides, I just wanted to just hone in on the sort of the first element of what we've been building, which is really an extension of the approaches LARVOL has been taking to public data for the past 17 years, which is applying both curation as well as text mining to take this broad universe of information and pull it together. So there's really two elements to this. One is what we can cover from a market perspective. So we can cover approvals and get alerts to our users that way. I also have a team that's specifically honed in on all the news, and there's so much movement these days in the predictive clinical diagnostics, companion and complementary testing spaces, as well as all the companies there. So we're covering that, and also doing some curation of, it's just such an important topic right now, curated news in ctDNA, MRD, and liquid biopsy. So this isn't, it's more elbow-deep manual sort of market assessment for competitive intelligence that we're pushing in what we call our digest. And this is available to get on a weekly basis, and also we can do specialized alerts. And then the other sort of side of that traditional, if you will, or core technology from LARVOL is the text mining. So here we sort of cover more by indication, and we're using a text mining specifically in Very to find the biomarker gene. So we're trying to keep our users abreast of all movements within a PD1 or P3 and RB. And some of those examples are here, and it can range. It can be trial updates from ct.gov or use your CT, or it could be reviews, case studies, or even preclinical. This is all pre the indexing I was in associations I was referring to earlier, but this is what we're doing with text mining to be able to tag this and organize it. So I wanted to sort of pause on that and see if there's any questions from the chat box, and also any questions or comments from my panelists, Karna and Vincent.
Yeah, one thing that I've found very valuable are these diagnostic news updates, especially with respect to anything that can get the NGS panel results back faster. That's one of the challenges that I've been seeing in the community oncology practices, where the results are taking weeks to come back. And so therapy decisions are being made before the results even come back. So that's very helpful, as well as these alerts.
And I've seen this, going through with my team, and then we, our primary user base right now is pharma. But I would imagine that the market that speaks to that is directly into the physicians, knowing things that can impact that speed of turnaround. So that's certainly something we cover, but also maybe haven't been thinking as much about that use case. I've been thinking more with the oncologist with respect to the heat maps, so I appreciate that.
Mark, I have a question for you. Is some kind of a new development? I think there was maybe a 3-day meeting in April from the FDA Oncologic Drug Advisory Committee talking about some dangling accelerated approval for checkpoint inhibitors. I'm not sure if people have heard about that. There were 10 indications that were in discussion because simply the confirmatory trial that were supposed to verify the data used to grant this initial accelerated approval were unfortunately not confirming that. And some of this accelerated approval has been withdrawn. Is this something that you're tracking with VI?
So it's an excellent point, and I don't think my team and I have fully wrapped our heads around all the different points of light that we'd like to cover with respect to that dangling approval. Now, if that dangling approval ultimately proves actionable and the lack of confirmatory data leads to a reversal and an approval that is most documentable in the heat map, and in the platform and the database. But I think that is a new type of news item that we haven't expressly been covered, that I'm taking up with the team from the news perspective. And then also from the indexing perspective, I think we should take a very careful look. And when it comes to what's inside the evidence stack, which I'll be introducing in the next slide or so, that we need to take a hard look at inserting that, because it's a recent development, especially because, as I didn't expressly say it, we cover as an alert acceleration, and we cover that as the news item. But if they sit and sit and sit, there's certainly something to be addressed about that. So thank you.
We have time later, maybe when you look over, you do a demonstration with it, you can maybe try, I think one of accelerated approval that's been withdrawn, I think is, for instance, is nivolumab for HCC. That was accelerated approval in 2017, but they decided to withdraw this indication. So we can maybe look at it if we have time at the end of the webinar.
Sure. And that was in hepatocellular?
Yes, HCC, yeah, HCC. Nivolumab in 20, that's the one that I can recall. There is another one where it was atezolizumab, the PD-L1 blocking antibody from Roche for TNBC in combination with NAB, paclitaxel. Here in this case, we decided to maintain it because there's really lack of option for TNBC patients. So even the confirmatory trials could not confirm the early trial, we decided to keep it as an example. Yeah, that it's kind of interesting.
We do, it's sort of not a perfect system, but we do work on curating and annotating non-supportive evidence and calling it out as such, even if not programmatically, but calling out in terms of the absorption that we do, that if something is counter, that we do try to put it in the context of any time where there's non-supportive evidence. So we'll definitely note that down.
I think that would be the kind of thing that would be nice to flag. For instance, to have I don't know, something flashy information somewhere in the window for people who are using and looking for information, just to because it's difficult to track everything. So I think this is a kind of very key information to be aware of. I think it could be very useful for doing that for people.
Absolutely.
Yeah.
Absolutely. So maybe to put so a little context about some of the things that have already been mentioned, how we index, how we create the associations, the evidence stack. I've sort of been pre-mentioning things that I guess I haven't verbally or visually introduced quite yet. So switching gears from that competitive intelligence focused text mining news push is what we've been doing with the very platform that we've been building, and we have, I think, a long way to go for, we hope, a variety of use cases from research to supporting oncologists and others, and in pharma, of course. So sort of three approaches that I've been honed in on is A, from a curation standpoint, which has already been sort of referenced, and that allows us to bring competitive intelligence to our pharma customers, as well as creating a research database. And that goes beyond the text mining. That involves a curation team indexing the data. And so we'll hone in on that maybe even with a demo. And then I'll be relatively brief on the other two sort of future directions, call them experimental or future direction, which is new ways to visualize the data, as well as some explorations and sort of prediction. That's, again, quite experimental, but we're looking at new ways to apply our data. So to sort of level set for a second and reintroduce some of the concepts I've already sort of mentioned, is in terms of curation, and these numbers could probably easily be updated after ASCO, which was a huge amount of information. We've covered thousands of genes and drugs, and over 150 tumor types when you look at the subtypes, of course, and the thousands of biomarkers. And just to be very brief, and I think this speaks to some of the questions about noise in the data, the very brief way to describe our directive is to document and index predictive response relationships, meaning at the level of the publication, we can pull out that T790 is predicting, or that non-small cell lung cancer with T790 and treated with that VEGF2 inhibitor with erlotinib shown is sensitive. So there's obviously a lot more information to be had about that predictive relationship, but that's what our curations team primary goal is, to put that in a database and make that findable and searchable. And as you can see on the right-hand side, we do the excerpt. So we link you to the publication or the abstract or whatever it may be, excerpt a little bit of the information. We attach it to the trial ID in UDRA or NCT, or CT.gov, excuse me, and try to put all that information there to be accessible as an exploratory tool or as an intelligence tool to even keep abreast of your competitors. And I'll pause after this slide, so I'll be very brief here, is obviously with a database, if you index without some sort of hierarchical structure, it's not going to get you too far. So what we're doing there is using the AMP and ASCO, well, and also College of American Pathology standards, to where evidence is indexed by its level. So what I mean by level is the highest level of evidence would be an approval. In the approval, in the label itself, there's a relationship between the drug, the indication, and the biomarker. And covering down to late trials, early trials, of course, we cover ongoing trials where predictive biomarkers are included in the inclusion criteria. And more recently, we've delved into case studies and preclinical as well. So we do constant surveillance of a variety of sources. Of course, your PubMeds, your CT.govs, your UDRA CTs. We also keep an eye on Twitter, other social media, and obviously conference coverage, as mentioned, because that's a huge part of the type of data we do. And here you can see labeled in red, this is an older slide, full disclosure, but we try to highlight that to our users, that all of the news coming out is in a particular day, that we can bring that to our pharma users and other users as well. I'm going to pause. That was a lot, but I wanted to make sure introducing the term so we can all be on the same page about what we're trying to accomplish with the platform. But maybe I'll pause for questions about levels of evidence, about how the data is excerpted, etc.
And so, Mark, how are you tracking the longitudinal aspect of this curated data? Like, what happens if the data starts to contradict each other, whether it be in a conference abstract or a trial update and what have you?
It is an excellent question, and what I'll say verbally, and then we'll jump to the the live platform in a second, is we do what I'll call a data stack. So when you think about a relationship between a particular indication, a particular biomarker, and a drug, especially in some of the most common drugs, that's not a one-off event. So what we're doing is whenever that relationship exists, we're trying to capture that. It's basically, I call it a stack, but it's basically in a column on the heat map. So all that information is put in one spot. So for certain relationships, you're going to have line of A1, A2, B. So we try to track over time from the approval backwards to the preclinical and the reverse. Now we're a new team, building in the past year, so we wouldn't claim to cover all the entire history of biomarkers and all the history of its study in cancer, but we do cover that stack that way. And I think, as I mentioned before, if a particular piece of evidence with an association contradicts the rest of the stack, we try to call that out and flag it in the excerpt that it's non-supported evidence. That's what we do in terms of longitudinal tracking.
I think we have to be aware, of course, sometimes, of course, it's science complicated because obviously we know some markers might work for some cancer type and do not as well for other cancer type. There's a lot of agent also combined with other drug, and sometimes I can change also the sensitivity. So that's why, of course, I like to use a heat map because you can see sometimes this is not just black and white story.
Exactly.
And I also do like the fact that you guys, because I know it was there at the beginning when you started with VRE, is that you have both the sensitivity and the resistance. I like the color scheme between the green and the red, because both are super important for trying to guide developer for selecting as best as they can the patient population.
Absolutely.
For an example, I know I have an example. For instance, it's like for the PD-L1 or PD-L1 blocking antibody, there is some data have not been maybe demonstrated at less stage, but like MDM2 amplification seems to create resistance. So that's an example. I like that I can see that in the heat map.
Absolutely. And I'll actually go ahead and do a quick change. Is now I want to move to the sort of next element, which is what we're trying to do with visualization. And the primary thing we've been building with visualization are these heat maps. And rather than go through the details on this slide, I'm going to go ahead and switch my share screen and jump over to Vary, and hopefully you can still see my screen. If Karna or Vincent, you could confirm for me.
Yes.
Excellent. So in the spirit of PD1, jumping over to Nevo. So here's the sort of the type of information I've already shown. What's at the sort of the top line of it? But when you drill down, you see more information as you move along. So in the context of Nevo, I'll show the heat map in a second, but just to jump into that concept of longitudinal tracking, here's just a random example that was second on the list. So you can see here that the relationship of PD-L1 expression in GEJ with Nevo, it's contextual. It obviously is not as simple as that sort of 3 points of light or 4 points of light. So what you see here is we've got a Lancet publication from recent. We've got ASCO 2020 data. We have AACR, excuse me, that was 2021. We have AACR 2021 data, and on as much as we can cover from both conferences and the literature. But this doesn't, I don't think there's a contradicting evidence in this stack to my knowledge, but this gives, and I hope I'm not scrolling too quickly and making people nauseous, this gives a flavor of what we're doing in terms of tracking that longitudinal evidence, and we try to give just enough that we can fit into the database to tell the story a little bit. And then I'll pause for questions from chat and others. This is the heat map that's been referenced a few times, and we're obviously on the Nevo page, so all of the data is associated to nivolumab. And as you can see here, we've covered, nivolumab being used in so many tumor types and a variety of evidence. That's across the top axis. And the left-hand axis are the particular biomarkers. And as I hope this illustrates a bit, maybe a bit small, is we cover a variety of different types of biomarkers, from expression to TMB, broad mutations, specific point mutations, and combination mutations such as this EGFR, ALK, and TMB high. And then our goal in the heat map is to give you a quick visual. You can jump around, you can search for particular biomarkers up here. For example, if you were to go to, let's say, well, how about this? So this is actually not the specific reference that you are looking at, Vincent, but I wanted to stick to the exploration there. That here with Opdivo is actually an ADAM9 study in hepato. So that a quick search there can pull the different tumor types together and just give you a quick view of what we can do. And then I said I'd pause, but I wanted to jump to just one more page because it's such an important development.
By the way, Mark, if you come back just one page before, I think when you had A1 or A2 approval for, oh, but sorry, this is nivolumab. Yeah, nivolumab HCC. Can you go back there? You had, I think, A1 or A1, A2 when you look for HCC.
Oh, sure. Let me research it real quick. Thanks, Vincent.
I'm just curious to see that.
Yeah, so we do. This is a little hard to.
Yeah. So this is, I think, if I'm correct, that I'm not because of course that depends which line, etc. But this is the one we need to look. Here we had time. This is the thing I would look for because is it the one that has been actually withdrawn recently? Because the confirmatory trial could not confirm. So is it a accelerated approval here? I'm not too sure if we can see clearly where it is indicated.
Sure. So I don't think I have that acceleration in this stack because this. So this is sort of a specialized treatment. It's really an important point that we're still sort of working on structuring. We wanted to build a predictive biomarker database where it's a measurable biomarker at quote-unquote baseline that can inform a treatment decision. But we realized so much pivots around approvals that may not have a particular biomarker driver, or maybe they were initially studied with a biomarker and then they ended up working across the board, which has happened multiple times with your PD-1 and your PD-L1 inhibitors. So this one we'll definitely investigate separately because I don't have the acceleration documented, but because I already had, my team, we had already documented that 2017 approval. We backtracked the data so you could see all the A2 guidelines, and we may want to revisit this in the context of the dangling accelerated approval. So that's an important catch, Vincent. Thank you.
What I would add, definitely what is great is that you're not just covering a trial and approvals of drugs that are paired with a companion diagnostic. You're also covering one drug, like in particular, like we all know with PD-L1 or PD-1 blocking antibody. Sometimes they are approved with PD-L1 expression with a companion diagnostic. Sometimes they are getting approved without. And you're covering both, which is, I think, very important. You have a full picture. So that's something definitely to highlight, which I like a lot too.
Well, we do our best. We mostly hone in on what we call, quote unquote, no biomarker, as you just described, on later levels of evidence, so late trials, approvals, and guidelines. We haven't endeavored to go all the way backwards to the historical preclinical case study data, et cetera, because we think the value add is actually in the reverse direction, having that pivot of information from the highest level. So that was sort of a strategic decision there. But I appreciate those questions. That was a great way to illustrate sort of the moving parts of what we do on the heat map and the visuals here. And I just wanted to jump real quickly to show that you can search the data in different contexts. So you can search for, if you wanted to see a huge heat map, you search for non-small cell lung cancer. If you want to see another semi-huge heat map, you look at a PD-1 or a PD-L1 inhibitor. But you can also jump to specific genes or specific biomarkers. And obviously, it's been such a huge, important development that KRAS G12C now being targetable in non-small. And so it's very much on top of mind. I wanted to jump to say you can also search and track development there. So of course, this approval that I was just referencing that we're going to have in the database. And for that matter, there is obviously the other data here tracking over time. In KRAS G12, we have a very long list of the work that's been done and is documented in our database over the years to explore G12C becoming that druggable target and a biomarker. So obviously we have a heat map for that. And I don't think we'll have time, but just to illustrate for all on the call, all in the virtual room, that if you're interested in TNBC, which you mentioned, obviously, Vincent, with Atezo, with nab-paclitaxel, I don't think we'll have time to dive into that situation, but just to illustrate, in triple-negative breast, you can look at a heat map under those auspices as well. And as in a subsequent slide, I'll talk maybe a little bit about ways to apply our data for research purposes in triple-negative breast. is something I'm interested personally as someone who studied triple-negative breast in my PhD. So let me pause again to see reactions from the chat or questions about, the buttons or the approaches that were taken, the heat map, before we move to the rest of the slides.
So how are you making some of these predictive associations in that kind of absence of like patient-rich data, especially when you start to look at some of those early preclinical or case studies in terms of trying to make those associations? Can you speak more about how you're modeling with these types of data sets?
Sure. And I think it would actually make perfect sense if we jump to the very predict angle before I just jump back to the sort of more experimental visualization. So we need a visual because it's a little hard to describe verbally. So let me jump and see if I can help answer your question over here. Now, you raise an incredibly important point, which is I'm fundamentally aware that the type of data, public data, that we do both from a news perspective as well as a curation perspective, that type of public data is fundamentally different and it's easily stated less rich than patient data. So one of the and I think I prefaced it this way, experimental things we're doing and working on continuously is rules-based algorithms to make predictions. So the approach we've taken is as follows, and it's quite, it needs a lot of refinement, it needs a lot of input, but this is the framework. So we created basically two cross-database matrices. One matrix, single, does tumor proximity. Now we've just really created a basic framework of tumor proximity, where if you look on a scale from 0 to 100, two different types of breast cancer or a lung adenocarcinoma and a non-small cell lung cancer would be more proximal than, let's say, an acute myeloid leukemia and a sarcoma. Now, those are sort of apocryphal examples, but that's the framework that we've tried to lay down. And I think over time we need a lot of input from specialists to inform the proximity between the two, because I perfectly am well aware that that's far too simple at this time. So that's what we're doing in terms of tumor proximity matrix. So that matrix feeds into the algorithm that we built with a really great engineering team in France. And we've also built what's slightly more sophisticated drug proximity matrix. So we've taken all the drugs in our database and we've created that same proximal value, 0 to 100 basically, based on A, mechanism of action, which would be self-evident. B, we're sort of using our own data internally, similar gene activity, meaning the drug has been used in the same context of different genes. And third, the drug has been used in the same context of the specific biomarker. And so all of that feeds into the rules-based algorithm to try to make predictions. And I won't get into all the statistics per se, but what's being shown on the right-hand side of the screen, and my apologies, I didn't switch back to full demo mode. Let's go here. Is in that top right cluster of red that you see, we actually compared the predictions to the empirical data in the system, and they line up fairly well. And we're continuing to improve this modeling, and on a weekly basis, and it would be a whole different panel discussion, but on a weekly basis, we're tracking how often does new evidence come into the system that matched a previously recorded prediction. And it's very experimental, and knowing a bit of your background, Karna, from a patient data perspective, I will yield that this is not as sophisticated as the models that are out there that maybe you're better informed about. What would be your comment to that, Karna?
Well, it's rarely a case of better or worse. You can only work with the data that you have. and make the best case that you can. I always have the, from a real world perspective, I have always have the benefit of looking backwards. My hindsight is always 20/20 when it comes to knowing the outcome and then being able to feed into the machine learning. So it's a very interesting approach. If I'm interpreting the graph on the right correctly, it appears that you do not have too many false positives, right? Which is a great thing to see.
We're still sort of parsing it out, but we did discover that there, just as you probably could see on the heat map, there's a bit of a dearth of resistant data compared to sensitive data, especially when this this readout was made, which is a little bit dated now. I'm working on a new readout, but it tilted the algorithm to be better at predicting sensitivity than resistance. And
very interesting.
It's hard. I don't want to overinterpret that, but I have some instinct that predicting resistance, I think, would be harder. Just from just a gut or former bench scientist perspective, that's logical to me. But it was a limitation just because we didn't have, I think, the same balance of sensitive and resistant data. But
no incentive to publish the resistant data. So
yeah, yes, the lack of incentivizing of the resistant data can sort of tilt the system. Now, we do get nice sensitive data from, more sensitive data, or excuse me, resistant data from case studies, but has already been queried in the chat box. The level of validation and is, I think, great for a searchable database, but it becomes, much more impactful questions when you're trying to feed a predictive algorithm. Now, that's not going to stop me personally or my team from pursuing this experimentally, but the question always arises, would a physician use this to make decisions? And full disclosure, we're not there yet. But as a tool, as a research tool for maybe pharma, CI, and things of that nature, I think that's where the use case is, and we'll continue to develop it. So I really appreciate you asking about the predictive modeling. Thanks, Scott.
And I will say there's an interesting question in the chat to the panelists. If you're flipping back to the heat map, Mark, is this heat map approach only for cancer?
So thank you for that question. Sorry, and thanks, Karna. I missed the question. Right now, it's a strategic decision that we are completely focused on cancer. So as a company, we're both in biomarkers as well as some other platforms we're building in clinical data, social media monitoring, a number of others. We're completely oncology focused. So I think in the near term, we're not going to be expanding at this time. Now, there could be some room for exploring precancerous syndromes. It might seem direct to do, so that might be a possibility. But I wouldn't foresee at this time getting into hematology, cardiovascular, and other states.
Mark, do you think that, can you promise that for any user they can find everything, anything related to anti-cancer drug for all kind of targeting agent? They could find all the information, or you guys are still building your database?
I've learned over the years to avoid making promises of 100% coverage. Here's how I'd answer the question. So A, we're certainly still building out the database. We're focused on move forward data, conference data, new publications now, and we have, backfill. Now, there's a more specific answer to that question, which is, do we want to expand our approach beyond predictive biomarkers? So let me try to rephrase what I mean by that. There is a lot of cancer drug data out there about especially new compounds, new agents being tested in a variety of indications. And there's, early phase trial data about the efficacy and proof of concept of that agent. Now, we haven't covered that entirety. What we've honed in on is if a measurable biomarker predicts response. And it's something, I think that framework works for the heat map, but one could make a strategic point that for covering news, which is where we started our discussion, that for covering news, could you potentially expand? Now, for that, I think I mentioned Pulse, but didn't describe it. We have tools at LARVOL which cover drugs and drug classes. And from a competitive intelligence standpoint, that would constantly feed you information based on products and brand new products. So it was an important strategic decision, and one that sometimes I revisit if we should be expanding further.
I would love that. Of course, you know that.
Well,
I have an example with, myeloproliferative disorders, which maybe, I don't know, if it was several years ago not considered as cancer, but things have changed now. It's now considered as cancer. And I would love, obviously, to be able to use Veri for looking for any treatment, cancer drug being developed for treating this type of indication.
Yeah, I can certainly understand. So, and we might be at a good, this is more of a strategy than a technology thing. There might be in the not so distant future a place where we could pivot because we've been able to build out so many different tumor types and being able to cover a lot of gene and biomarker ground to date. So, maybe that's not as far away as it once was, maybe even 6 months ago. Yeah. Thank you, Vincent. So, I'm checking the chats. I'd like to switch. So we've looked at the heat map, and I hope we've given a sense of that. And I hope I don't want to forget to mention. So for research purposes, you can go to veri.larvol.com, and you can cruise around. You can also, if you want to see that evidence detail, the excerpting and all of that, you can create your own account for free. So I just wanted to mention that if I neglect at the end. Great way to get experience with the heat map if you're interested for just to research. I do want to mention one more thing because it speaks to sort of some strategic directions that we're taking at LARVOL that we're a completely remote company and always have been. I've never had an office. So we've actually been transitioning to use virtual reality as part of our office, but I've taken that as an opportunity to use virtual reality to do data visualization. So we created a partnership with the Caltech group called Virtualytics, and I've been working with them to basically create visualization tools for heat maps that can be explored both in 2D but also with a headset on in 3D. So I'm just using a screenshot because it's a little bit hard to show the full software. But the very front panel of what you're looking at is a non-small cell lung cancer heat map with the usual colors and letters as already shown. But what I've done is, with the Virtualytics team, is stacked it against a number of other tumor types along the backward depth axis in a cube. And without really having the time to go into all of the details, we're really excited to be able to use 3D visualization as ways to look across indication, see more data in one space, and hope there's applications here, both the standing applications for our pharma customers, as well as new ways for researchers, physicians, and others to get a sense of our public. And I'll pause there for any comments from my panelists, Vincent or Karna, on that matter. And then I want to leave the last 5 minutes to cover any questions in the chat box.
So Mark, how do your customers interact with this 3D? Do they need their own special headset? I mean, how does this work?
Yeah, so for customers and advisors, we work out providing a headset. So we use the Quest 2 We use the Quest 2 platform and provide that. And then if you have the Virtualytics software, you can get it on your desktop and then you pair it with the headset. And so how we meet with our customers and advisors and others involved in the project is you actually meet in a 3D data room. So you're all there in that virtual reality space with this cube. And the cube can be spun and swam through very Minority Report inspired, if you all recall that movie. That's how you can interact with the data. So really exciting exploration, and also the Virtualytics team, continuing to work with them, has actually built AI and more sophisticated analysis into their software. Not only are they a visualization team, they're also, it's not just about 3D. So we're continuing to work together to think about new ways of looking at the data.
That's kind of exciting. I mean, in my regular job, I use the headset for structure-based design. There's a company named Nanome where you can do the same thing, where people from different locations, they put the headset and they look for design. So they see from X-ray structure, they see the target, the ligand, and they can discuss in the virtual room and making chemical modification to the ligand, etc. This is fantastic. So I could imagine to do that with Viri as well. That would be that's kind of cool.
Yeah, so that's a little bit more sophisticated than I guess the cube we're looking at. But there's just so many possibilities here, and we're just excited as a company to be exploring this for data and also exploring it as part of our work environment using VR. It's a great way. We've all been quarantined for a year, and it's been such a wild year. It's been interesting to explore new ways to interact. So before I conclude, let me check the chat box again, because I do think we're coming up on time. I see, do we cover biomarkers of extracellular matrix? I don't know if I could answer that question specifically. We do not preclude that if, let's say, a preclinical study showed increased levels of an extracellular matrix protein, or let's say a protease, we do not exclude it, but it's much harder to find predictive data for an extracellular matrix protein. It's something I should probably revisit a bit. It's an excellent question. I personally studied plasminogen and MMPs and plasminogen inhibitors and thrombin and how they degrade the matrix for a number of years. So it's a point of interest. So excellent question. Definitely something to revisit. So let me take the last minute to thank everyone for joining today. We had some great questions from the chat. I'd like to thank Vincent and Karna for their insights. Appreciate you joining, gentlemen. I'd like to thank Elizabeth Lamb for fantastic organization of today's event. And maybe I'll conclude with, here is where you can check out Veri.com, larvolveri.com. Also some information just to leave up on the screen where you could contact us or me. So that's me at the bottom. And obviously, if there are subsequent questions or time here together, we'll do our best to answer them offline.